Good afternoon to the audience. My name is Jean-Yves Maillard. I'll, the, I'm the, I've got the pleasure to be the host of this River Teleclass Education today. And I've got the pleasure to introduce a colleague of mine, Dr. Matt Garvey. But before I do that, this is a A.D. Russell Memorial Lecture. So Denver Russells, who passed away now 20 years ago, was a pioneer really in pharmaceutical microbiology and especially in disinfections and control of microorganisms. He wrote over 400 publications, several books, and it was one of the world, or if not the world expert on disinfection at the time. And I'm very, very honored to be able to host this very specific or special teleclass educations. So a few words about my colleague, Dr. Garvey, which I'm very, very pleased that he's accepted to give tonight's presentation. Mark has worked in the field of infection control for, I think, over 9 years, maybe more. You can correct me later on. He's a consultant clinical scientist in microbiology and is currently the Deputy Director of Infection Prevention and Control for the Infection Prevention and Control Services at University Hospitals Birmingham, NHS Foundation Trust in the UK. Mark is also the director of the Hospital Infection Research Laboratory, with the main function to investigate hospital infection, providing practical advice to health service professionals and the industry throughout the UK. I believe he has more than 40 publications, all related to infection control, including to some of the most common peer-reviewed journals or academic journals in the field, whether UK journals or US journals. Mark has a current research interests include water microbiology, focusing on Pseudomonas aeruginosa, Mycobacterium chimera, general infection prevention and control issues from MRSA to carbapenemase, producing Enterobacteriaceae. We are very, very fortunate to have Mark because usually it's very, very difficult to hold to contact. He's always so busy. So I'm very, very happy and pleased to have Mark with us tonight. Mark, if you'd like to start your presentation.
Thank you, Johnny. Yes, I probably need to update the bio a little bit. It's a slightly older one, so it sounds better that I've only worked in IP for 9 years. I think it's been a bit longer than that. I think I started back in 2009, 2010. So that's probably a few more years and a few more publications.
I thought so, yeah.
Yeah. Whoops, that's my bad on that. So yeah, I've got the. Thank you for asking me to present today. So it's a great honour to talk about some of my work, and it's really nice to get asked to talk about one of my favourite subjects, what is water microbiology. So that was some of my first pieces of work in infection control. It's later progressed a little bit. So yeah, I'm the Clinical Director of Infection Prevention Control at University Hospitals Birmingham. There's a few disclosures, but nothing huge amounts. Just done some expert panels and some sponsored talks at various different places, but I've not received any money myself for anything that I've done. So this is a picture of the hospitals that I work at in the UK. In essence, I work in a city called Birmingham in the UK. Birmingham's right in the middle of England, and it's the second largest city in the UK. So my job, I'm responsible for infection control at a conglomerate of hospitals in Birmingham, and so these are situated all throughout the city. So I'm pretty much in charge of all the hospitals across Birmingham, apart from one over the west of Birmingham. So we have in the yellow, we have a big hospital called the Queen Elizabeth in Birmingham. That's our UK military hospital as well. That is basically it's a nice new building that we moved into back in 2010, but we've also moved back into our old hospital due to capacity reasons, and then we built a new private hospital in between that. So that combined bed size of that hospital is about 1,500 1,500 beds. The QE is very, very different. It's like a tertiary referral hospital in the UK. So, for example, if local hospitals have an issue with, like, say, can't haven't got the speciality to care for a patient with like neurosurgery or what have you, they come into the QE to have the specialized care, and then they go back to their local hospitals. So the QE is like a specialist hospital for various different things. It's a major trauma center. It's the leading Centre for Solid Organ Transplants in Europe, and does the most amount of solid organ transplants in Europe. So, as you can see, a very specialised hospital, and pretty much encompasses everything. Then we have other hospitals across Birmingham. We have the QE up in, so we have Good Hope Hospital up in the north of Birmingham. What's the blue picture there? And that's like a district general hospital, a small hospital of about 500 beds. Then we have Heartlands Hospital, what's the green one, over at the east side of Birmingham, and that's about 700 beds. And then we have a Solihull Hospital again over the east side of Birmingham, it's more like an elective hospital, but it has got like a bit of emergency capacity in there. Now, Birmingham has a very diverse population, and has a lot of has a diverse ethnic population as well, and it has a lot of social deprivation, probably one of the highest social deprivations in the UK. So Birmingham as a whole sees a lot of infection, and as a result we get a lot of infection through our hospitals. So again, that's just a little bit of overview of our hospitals. The QE is where I'm going to talk about a lot of the water work that we have done. So let me skip over. This is an overview of the slides. Mainly I'm going to talk about water and the issues of water, a little more focus on Pseudomonas aeruginosa, but we'll talk a little bit about other infections. Pseudomonas aeruginosa, everybody in the UK, this is my favourite organism actually. Why I like it, well, you shouldn't do this nowadays, but it's the smell and what it looks like under the, it smells very like caramel-like, and when you look at it on an agar plate, it's very gloopy, very has a nice colour to it as well. So that's probably one of the reasons I like it. Also, it has one of the largest genomes, and it is an environmental bug, and it's very cunning, and it can become easily resistant to antimicrobials due to its efflux things and efflux pumps and so forth. So it's one of my favourite bugs. So it's widespread in the environment, and in hospitals we can often see it in our hospital water and domestic sinks and what have you. And then when you look at like human infections as well, humans tend to get infections, or they tend to be colonized, it tends to be at moist sites, and it's very highly opportunistic pathogen. Now, back in 2010, when I first started in infection control, I was actually down in Bristol. Again, another reason why I like Pseudomonas aeruginosa, and there was a series of hospital outbreaks, mainly in the neonatal units with Pseudomonas aeruginosa, and some of the sources of those infections were associated with water. So that's why I'm going to talk a little bit about water, and why I'm going to start with Pseudomonas aeruginosa. I won't go through this in huge amounts of detail. This was pinched off a colleague of mine, Mark Webber, many years ago, but Pseudomonas can form some very nice biofilms, and we know what probably a biofilm is, and that's just a nice schematic of how when you look at like a tap, you can get, like, if you've got Pseudomonas there, it can attach to a surface, it can mature, and then obviously you get dissemination of bugs. And I don't know if anyone's ever looked down a shower head, taken a shower head off, and you can see slime down the flexible hose, for example, and that tends to be a biofilm, like plaque on your teeth as well, or if you go out walking and you see slime on a rock, biofilm, and it's a city of organisms, and Pseudomonas is very good at making biofilms. And this is just this is actually taken from my hospital. So this is a swan neck tap that's in our new QE hospitals. These taps are pretty defunct now, but hopefully you can see that green slime around the tap, and some of the in those pictures. And again, that is a biofilm of organisms, and we have cultured Pseudomonas aeruginosa from there, amongst other things. So you can see on your taps, you can get bugs and growth of bugs on there. So we know we can find it on our taps in our hospitals. So I'm talking a little bit about Pseudomonas and the role of water microbiology, and obviously it's garnered a lot of interest. It's garnered a lot of interest in the UK of late, even more so back in 2010. But actually, this is not actually anything new. So I'm fortunate to be the director of the Hospital Infection Research Lab. What was run by the great late Graham Ailiff as well, and he's done some publications on this. And this is just some old papers back in the 1970s showed that Pseudomonas could be found in sinks and taps, and you can get splashes with Pseudomonas aeruginosa from these, and transmission of organisms. So it's not a new thing. We've known about it for many years. So this is when I was back in Bristol, and this garnered a lot of interest in the UK and got a lot of media attention. It even got some police involvement. So there's a series of baby deaths with Pseudomonas aeruginosa, and when our UK HSA was one of our governing bodies, investigated it. They found the source of the babies getting Pseudomonas aeruginosa was from the water, and there was lots of work and lots of media attention in Belfast and in Bristol around that. So when babies are born, I should say, when babies are born, they won't actually be colonized with Pseudomonas aeruginosa. So when you get neonates that get infections or colonizations with Pseudomonas aeruginosa, you actually automatically think, well, where have they got it if they've not been at home? And so that's why it probably leads you to think, well, let's look at the water, let's look at the sources of that, and then that's where you can get it. An adult is very different. An adult could be colonized with Pseudomonas aeruginosa. So as a result of these neonatal outbreaks, we had some not great guidance, but we had a health technical memorandum looking at giving us advice on how to monitor and look for Pseudomonas and what we should do, how to control water microbiology such as Legionella and Pseudomonas in our healthcare settings, because it garnered a lot of interest. It talked about having a water safety group, what involves the state's infection control and relevant colleagues, and then talked about regular testing of taps for Pseudomonas aeruginosa in augmented care areas, so like your intensive care units or like your transplant areas or hematology, and do that kind of testing. So that came out. So when I came, so I was down in Bristol, did a lot of work around Pseudomonas aeruginosa. So then I moved to University Hospitals Birmingham back in, I want to say 2010, 2011. And obviously I was very interested in Pseudomonas aeruginosa. So one of the first pieces of work when I moved into infection control was I wanted to look a little bit around Pseudomonas. Now, at the Queen Elizabeth Hospital, we have got a 100-bedded co-located critical care unit, what's like the largest co-located critical care in Europe, and at the time it was probably the world. And one of the units was our trauma unit, and it had like burns critical care shock rooms on there. Now, I noticed from my job as like the job I was doing at the time, infection control practitioner, I noticed that over a period of time, I saw an increase up in Pseudomonas aeruginosa cases in our patients that occupied these burn shock rooms. So this is all in that paper I've just presented on the slide. But what this shows is in this one burns shock room, we had 5 successive patients that went through that room, and they all had clinical specimens that grew Pseudomonas aeruginosa. So all 5 patients that went through this shock room, had like major burns injuries. When you go to these shock rooms, you have well over 60% total burn total burns across the skin, and burns patients shed a lot of microorganisms in the environment. Anyway, we had these 5 patients that went through there, and as you can see, they're admitted. And obviously, you take a lot of swabs from their burns wounds anyway, and as you can see, they all grew Pseudomonas aeruginosa. So we did a bit of typing, some whole genome sequencing at the time. At the end of this, and we found that all 5 patients had exactly the same strain of Pseudomonas aeruginosa. And I was like, okay, let's have a look at our water testing results. So we test the water in our augmented care areas every 6 months, as I alluded to. And what I actually found was that Pseudomonas aeruginosa was actually found from one of the hand wash basins right at the beginning of the very first patient. We managed to get those strains of Pseudomonas aeruginosa, and we actually found it was exactly the same as the patients. We do, when we have like a positive water, we do remedial work on those outlets, and we did remedial work of those outlets. I'll come to in a bit more detail about how good the remedial work is. So you did some remedial work, you take some more water samples, find there's no Pseudomonas aeruginosa, but actually 6 months down the line, well, it's not 6 months, but about several months down the line, we again found Pseudomonas aeruginosa in there. So again, and that we cultured that strain, and it was exactly the same as the patients. So again, we found a bit of, we knew that our patients had exactly the same strain, and we found Pseudomonas aeruginosa in the water as well. So that led me to apply for a small pot of money, what was about £100,000 at the time, to the Department of Health to look at the role of what is actually the role of Pseudomonas aeruginosa in our adult augmented care settings. You know, do we see transmission from our water to our patients? So again, if you remember when I talked at the beginning, neonates, when they're born, they're not colonized with Pseudomonas. So if they get Pseudomonas, okay, they must have got it from somewhere, but adults can be colonized. So this was a very simple study. It was over 6 months. We got money to sample all of our outlets on our co-located critical care units. So this, what shocked me, because I did all the water sampling, there was 231 water outlets on our 100-bedded critical care unit. We took water samples and had a look to see if we could grow Pseudomonas from those, and we did it at day like 1. We did it right in the middle of the study and right at the end of the study, just to get a longitudinal picture of that. And if we grew Pseudomonas aeruginosa from that water, we did whole genome sequencing on it. So very simple. At the same time, during that study period, that 6 months, we took anyone who had clinical isolates, we grabbed those, and we did whole genome sequencing on those, so we could see if there's a link between our water, if our water strains matched our patient strains. So very, very simple. So again, I won't bore you, but this really shocked me when we looked at the our water testing. At any one time, 30% of our taps actually had Pseudomonas aeruginosa in the water. We found it. That shocked me, the amount of it. There's reasons for that, because the Queen Elizabeth is, like, you remember when I talked at the very beginning, I gave a lot of history about the Queen Elizabeth. It's a new hospital. It was built in 2010. These were before some of our health building notes came in. So like the taps were water tested before we even installed them. We might have not undertaken the correct flushing, chlorination, and so forth. So there's a lot of reasons that compounded why we saw Pseudomonas aeruginosa in our tap outlets. And I'll come on to some of those other holistic pictures as well, such as like how we clean the taps, how we do like the remedial work. What they used to do, if you find Pseudomonas aeruginosa in a tap, they used to take it out, take it off the sink, they put it into a descaler, a bucket of Showerhead Plus, put the tap back on, and then use that bucket to go around to other taps that they thought were contaminated, and again do the same type of remedial work. And again, you can see how Pseudomonas spreads about. You also look at like human factors as well, although it's called a hand wash basin. Clinical staff are very good at putting things down the sink, what's not shouldn't go down there, like, you know, feeds, antibiotics, things like that. So there's lots of different things when we wash patients, putting the wastewater down there. So there's a reasons why we see what we do, the amount. So these are the results. So this is just a whole genome sequencing dendrogram, what was done in conjunction with the University of Birmingham. So that's Professor Nick Lohman's group. It was a multi-centered study, but I'm just going to talk about the Queen Elizabeth results. And the Queen Elizabeth results, so the water results are yellow and the patients are blue. I can't, or it might be vice versa, but the yellow and blue. But what you can see from this big dendrogram is there's a variety of different Pseudomonas strains. So sequence type 395 is very different to cluster D and what have you. So we see a variety of different strains. So it's not just one strain we see throughout there. There's loads of different strains, and those are the reasons we see loads of different strains, of because we've imported different strains into the hospital over the years. We've done testing at the source of when water comes in and we don't see Pseudomonas aeruginosa, and we don't see Pseudomonas aeruginosa in our pipes pipe work either. So it's really focused at the tap. So when you look at the whole genome sequencing data from our water and our patient isolates, what shocked me is about 60% of the water isolates actually matched our patient isolates. What's massive really when you look at it. So we know we definitely get transmission of Pseudomonas aeruginosa from our water to our patients, and our patients can contaminate the water as well. So lots of different things why we see what we did. So again, we didn't leave it like that. Again, this is a few years old now. So this just talks. This is probably the best paper. What talks about the cluster of interventions that we did at the time. So these are the different pieces of work that we did. So it's a timeline of different things over a couple of years. So we put point of use filters in some of the outlets. So you remember when I talked about those 5 patients at the beginning in that Burns critical care shock room. So one of the interventions we did, because we saw the remedial work of disinfecting tap wasn't great, we put point of use filters on all those taps. So that intervention's that one what talks to that first paper. Then obviously we did the Department of Health piece, point of work, when we found out we had that amount of transmission and the amount of taps colonized with Pseudomonas aeruginosa. We put filters on those, and then we did a series of other interventions. So we did holistic interventions, such as we trained staff not to dispose of water down the hand wash basin and take it to the sluice, or down the toilet, or down the drains of the showers, that kind of stuff, but mainly through the sluice. We used gel sheets to solidify the water as well and put it in the sluice. We also did removed all the taps, because the taps outlets, these swan necked ones, are probably not fit for purpose, and so forth. So we did various different interventions across the whole patch. This is just a breakpoint analysis, what was in that paper to a couple of slides back, talking about all the interventions, and obviously where you get the dotted and a breakpoint analysis, just the dotted lines just show you where something, when you put a change in, shows if it's statistically relevant or not. So these are some of the statistical analyses we did. We didn't use multiple logistic regressions now on that, but anyway, so that intervention is the filters on that on our Burns shock critical care Burns shock room. I should say that the y-axis, like the rate of Pseudomonas in critical care, the numbers of colonizations, and then the data on the bottom. So you can see the PAL filters had a bit of a drop down in Pseudomonas aeruginosa infections. Then when we put the holistic interventions in place, so disposal of wastewater and how we cleaned the taps, things like that, we saw a big reduction in down of Pseudomonas aeruginosa as well. So how we acted on our human factors reduced it down, and then obviously when we put new taps in, that reduced it down. So you can see we did various different interventions, and each single intervention reduced it right down. We've published different bits on those interventions as well. So this is just it's a small letter in the Journal of Hospital Infection, just to look at how you can clean a tap. I don't know if anyone has tried to clean a tap, but I tried to clean it by following our manual at the time, and all I did was put some UV gel, so you know the glow gel that sometimes you use for your hands, the UV glow gel, and I just put it into a sink, and I thought, well, I'll follow the manual and I'll try and clean the sink how one of our domestic staff would, our facility staff would do it. I don't know if you can see that top right hand, it's on my screen, right hand picture, but you can see the UV cream on the tap tip, and you can see I managed to spread the UV cream everywhere on the sink, and if you go to that paper, you will see it. Maybe that tells you one thing of me not cleaning your sinks in a hospital. Anyway, we came up with a different way of cleaning. People have come up with those subsequently, of like you having a separate cloth to clean the tap, the sink, and so forth. And then you can show that you can reduce down contamination. Education on that works. So that's another thing. Like I said at the beginning, we put filters on everywhere, so we think, oh yeah, we've got that's great. We've put point of use filters on there, but they come with their own problems as well. You're sort of creating a bit of a dead leg because you're not getting rid of the problem. And if you understand, you can get different plumbings in hospitals, and you will know that, or your and plumbers will know that, and engineers will know that, and you can you know how our plumbing works. It's like sucked from one tap to another, so you could easily create a dead leg on there. You know, when you have filters on, they're expensive. You have to replace them either like every month or every 3 months, depending what you've got. Staff have to remember to do that. They may remember it, they may not. And on top of that, if you leave filters for long periods of time, so this is again just another letter. The picture's not particularly great, but you might see on the right hand side, it looks a bit slimy on the bottom of that filter. And actually, what we did there was we saw that filter had been on for a while. It was slimy, and we took a swab. And we managed to grow Pseudomonas aeruginosa from that. So just because you've got filters on there doesn't mean you're getting rid of the problems at the time. The strain of Pseudomonas on there was exactly the same as the patient's strain of Pseudomonas aeruginosa in that bed space. So again, that doesn't always solve the problem. Again, holistic factors play a role in that. Again, this is another paper after that when we looked at like putting new taps in. So I'm not going to go through the taps designs. These are just some taps, but again, we looked at could you put some other taps on there. And so you know, taps will, when you look at water and take water, you will obviously get TBCs in water, and you will get biofilms. But we had a look at what type of taps you could put in. Could you put taps in that you could decontaminate? So this paper just talks about some taps that we got in that you could remove if you found bugs in there, or you found Pseudomonas aeruginosa in there, you could take them off. And you could put them through a thermal washer disinfector, remove the TMV and decontaminate them, or put them through an autoclave and put them back on. So others, you know, once they find Pseudomonas aeruginosa in the taps, they probably remove it and put a new tap in, but you can do decontamination as well. So there's lots of different things you can do to do that. That becomes technically tricky when you're relying on your estates to do it. And there's new tap designs out there all the time, and the taps are not water tested when they come in. So there's lots of different things in there as well. And this is just a schematic, another breakpoint analysis. You can see it's sort of the same time period as the other ones. This is just rate of Pseudomonas aeruginosa infections on there, and then when you put, and this is just on one critical care unit at the time, not like all of them. And when we put new taps on, you actually reduce down Pseudomonas aeruginosa again. So you can see how you can reduce Pseudomonas aeruginosa with like various different interventions. So I've talked a lot about an intensive care unit and the adult intensive care unit at about testing, but what about other high risk areas? So such as like we've got a burns unit. What about the role of Pseudomonas aeruginosa? Do we get water transmission to our patients in our burns patients? You know, I've shown it, we've had it in our burns critical care shock rooms with high degree percent burns. What about hematology? What about hemodialysis? What about solid organ transplants and so forth? So again, not an article in Journal of Hospital Infection, but in AGIC. So this just talks a little bit around transmission of Pseudomonas aeruginosa from water to our hematology cohort of patients. So this was an interesting little outbreak So, working clinically, we picked up that I think it was in 1 month. Was it in 1 month or a couple of weeks? I can't remember. But anyway, in a short period of time, we noticed that 3 patients on one of our hematology wards, our like stem cell transplant ward, 3 patients in like 2 weeks had Pseudomonas aeruginosa line infections. But what was really odd, they had central line infections, not peripheral ones. So we managed to go Pseudomonas out the central line. When we took peripheral ones, the peripheral ones were negative. So we thought, well, that's interesting. And then when we looked, they're all like in successive bed spaces, and I was thinking, well, that's strange. Have we transmitted Pseudomonas from one patient to another patient to another patient? Bear in mind, in our hematology unit, we've got point of use filters on all our taps. So I was thinking, well, this is interesting. I wonder how we've got Pseudomonas and aeruginosa have these patients acquired it. So we did some whole genome sequence again, and we found that all 3 patients had the same strain. So we went up to the unit. I went up to the unit, and actually I thought, well, let's have a look. Let's go and have a look around. And virtually everywhere had filters on. I was thinking, well, that's interesting. Then I went into the IV prep room where we make IV drugs, and I thought, oh, there's that. And as you can see in the picture, there's one tap that actually didn't have a filter on. I was like, oh, that's interesting. So that sparked my interest. So I thought, you know what, I'll sneaky microbiologist. I'll take a little bit of water from that tap outlet, see if I could grow anything from that. I noticed there's these blue trays. So these blue trays we use to transport our IV drugs to our patients and give the patients the IV drugs. And I noticed some of these blue trays were in the sink as well. So I thought, well, I'll take some swabs of those because they've got water in there from that tap. And to my, well, I suppose I wasn't surprised. I managed to grow Pseudomonas aeruginosa from the water and from the water droplets in the blue trays. So we'd sequenced those as well, and they were exactly the same strain as our patient strain. So what would immediately identified the source as this tap outlet, and what we'd done is obviously all those 3 patients were on IV drugs. We'd taken these blue trays, gone and done some IV work on them, and that's how they got the Pseudomonas aeruginosa infection. So again, you can see how holistic factors, human factors, and how we can spread Pseudomonas aeruginosa to our patients as well in a hematology. This was some work that we did with, again, Nick Lohman on our Burns unit. This is a really complicated diagram. It looks complicated. It's not. It's really not. It shows exactly the same thing. So the little boxes are just patients, and we've clustered like this the strains of Pseudomonas aeruginosa, and the different colours are just like, so blues, some water positive Pseudomonas aeruginosa. So that's Pseudomonas cultured from the water. Red is from like a wound swab where we've cultured Pseudomonas aeruginosa. The green is from the environment, and some other clinical specimens like pink sputum, yellow for urine. And as you can see, if you look at like box 4 as it's right in the middle, you can see we have a patient. We've managed to, and this box shows that bugs, the Pseudomonas strains that we've got are identical. So we found Pseudomonas aeruginosa in the water, we found it in the wound swabs, and we found it in the environment. And that box, all those strains correlate to be the same. So again, we found Pseudomonas everywhere. And then you look at, box 5, again, you found water samples, environmental samples, and clinical samples with Pseudomonas aeruginosa, and 5 is very different to 4. So again, we've shown transmission with Pseudomonas aeruginosa there as well. Not only that, what's a little bit more scary, in the UK, to control our water microbiology, we chlorinate our water. We have chlorination systems. We use a lot of chlorine to chlorinate it. From a piece of work that we did when we sampled did some sequencing on all our water isolates, we actually found some Pseudomonas aeruginosa strains that actually had a plasmid that we named P Burns 1 that actually had a chlorate dismutase gene in what it made it so it was less affected by exposure to chlorine. And when we did some work, biofilm work done at the university, again with Chris Thomas, and we exposed that biofilm with the plasmid into chlorine. Obviously it wasn't affected compared to some of the other cells that didn't have these plasmids in. So again, some interesting work as well on that. So we even find some of our main controls for water control doesn't get rid of Pseudomonas aeruginosa. So that's a little bit around Pseudomonas aeruginosa. I've talked a lot around that. So for the final 15 odd slides, I'm going to talk about a few other water issues a little bit. So I don't know if you probably experience like us in the UK. Staff can put things down the toilets and the macerators, like wipes and what have you, and the toilets and pipes can get blocked. I don't know if you can see that picture. That is actually a picture of a man trying to unblock a drain outside our hospital that's got blocked with like wipes and what have you. And that really is somebody that I took of a picture going to unblock it rather than him than me. But you can see we can get floods and blockages. This was originally a video on the next slide, but it just shows you. I was going to show you a video of a mass leak that we had from like a blockage, what completely put water. We had like reams and reams of water and burst pipes as a result of it. And again, the cleanup after that was massive. But again, these are some of our pictures over our other sites where you can see you get blocked toilets, blocked drains and sewers, macerators, and that can cause like, I've gone onto a unit where you've got loads of wastewater that's everywhere. And there's loads of articles out there. This is just a Spanish or Portuguese study. I can't remember. I think it's Spanish in Seville. Study that showed when you get like leakages and blockages and wastewater spills out into the environment, you can get outbreaks of like those nasty organisms, your carbapenemase producing intrabacteriacin, so forth. That's the video what I was going to play. So this, you can see how you can get a blockage and how look at the amount of reams of water that are coming from there. I've actually was in there in my wellies, and when I span round, you can see the whole corridor being flooded with with water and wastewater. So again, those are some of the challenges, and from an infection control point of view, you're going like, oh, they've picked up the phone, Mark, what can we do? I was like, well, obviously stop it, let's unblock it, let's clean it. Simple things, and try and get to the bottom of that. So I wanted to talk a little bit about water dispersal next. Again, this is just another, this was another video, but Paul very kindly put the different slides on there, and it just shows you a water droplet dropping. So if you go and look at that paper by Estrada Perez, there's a video in there, and it just shows a water droplet. So if you as you can see, a water droplet dropping down. So just imagine this is a water droplet, and just say that water droplet had Pseudomonas aeruginosa in, for example. And when you can see it dropping down, when it hits the surface, can you see those these little like dots of spray going off? So you can see how far water can splash. So if we go back to the first one, this is just some paper towel on the floor, and you can see how far water can splash about. When you go to when I go to the toilet and then wash my hands afterwards, I tend to get water all over me. Not because I've wet myself, it's because I've put water everywhere. But again, that just gives you an overview. And even if you think if you think of this water droplet, even if it hasn't got Pseudomonas aeruginosa, just imagine if that hit a sink or a drain that had got like microorganisms on there, you could get like spread of those organisms very, very easily from that sink. This again is just some more work from Estrada Perez. I quite like this, but again, it just shows you how. I just wanted to illustrate how far water can splash about. So this is just a mannequin having water sprayed on them, and the water's got fluorescent dye in, and you can see it spreads a variety around the area. And again, when you look at this, I just wanted to. I'm talking about sinks and disposal of wastewater, but like this is actually a clinical. This is our actual clinical setting. Look what we've got in these hand wash basins, and again, it just shows you like the taps leaking and what have you. So if you're getting splashing and you've got loads of equipment around, and you've got equipment within the sink, a lot more needs to be done. We don't actually think of like, oh, the sink is a risk factor. What actually do we do from a decontamination of a sink? When we do a terminal clean of a room, we don't do huge amounts either. I don't know if there's lots of stuff out there looking about what is down your sink. So look at some of these gruesome pictures. One of my colleagues hates these pictures. Some of these are from my hospital. That picture there is not, but these are from there. And biofilm grows very, very quickly, as Jean-Yves will tell you. It grows rapidly, like a millimeter every, in a short period of time. So what's down those sinks? It's like horrible organic matter, and we've cause a little bit of that of what we put down the sinks and so forth. But actually, when you look at like this top right hand picture is an IV. This is another hand wash basin in one of our IV prep rooms, and you can see it's not draining very well. And again, another one of these IV trays that we transport bugs in there. So me walking about, I was like, oh, okay, let me take a bit of that water and let me see what I can culture from this. And actually, I cultured, this is some chromogenic media, and that's actually a carbapenemase producing Enterobacter It's an Oxa48 Enterobacter cloacae on that plate. We actually on that at the time on the ward had an outbreak of Oxa48 Enterobacter cloacae on the ward. So again, you can see, from that wastewater, we've got a CPE down there, an IV trolley, and we've got transmission of that. So again, I'm talking about other organisms as well. And a couple of years back, we went a bit further and created a sink splash zone. And all we did was that when she showed you that picture of that water disposal, but all we did simply was put paper towel on the floor and ran a tap to see how far. So this again is this critical care burns shock room I was telling you about. Remember when I told you at the very beginning when those 5 patients had Pseudomonas aeruginosa? So we just ran the tap in a sink, paper towel on the floor, and we just see how far the water could splash. We found it up. You can see those black circles. We found it up to 2 meters away. It's remarkable, really. When you wash your hands or do something around the sink, you remember before when I said when I go to the toilet, splash water about, but look how much water you splash about. So if you've got something down the wastewater with water splashing on it, splashing out of the sink, you can see how much you can be splashing out of organisms. You've got Pseudomonas aeruginosa in the water in the tap, that's splashing out, or what it's splashing onto, lots of different things. On the left-hand side, this is a typical bed space in our critical care unit. So this is obviously a computer, but behind that is a patient bed space, so a patient will be there. Look at your hand wash basin. Look what is round that hand wash basins. That trolley there is an IV prep trolley. So within 2 meters, you've got the patient within 2 meters. You've got an IV trolley within 2 meters. The patient will have loads of devices ventilated and so forth. You've got PPE around there. So if you've got Pseudomonas aeruginosa in that tap, or if you've got something down that sink that you've never done decontamination about it, you could get various different things splashing onto that area and those patients acquiring it. So when I talked a bit about like how, if we've got Pseudomonas aeruginosa in the tap, how do our patients get it? Well, there's lots of different ways that they can get it, and the sink splash zone is one of those. We've carried on that work. Again, we've got to write this up, so this will hopefully be out in a few weeks' time. And what I've got in my lab is a handheld air sampler. So we've just taken this one step further. So what we've done is we've run the tap again on our critical care unit and just left this hand sampler handheld air sampler running, and it basically just sucks air in at a certain speed onto a petri dish. And we just thought, well, let's see what grows. And you can see those plates, got loads of different organisms on there growing. And what we found, to our surprise, we found like Citrobacter, Enterobacter, Pseudomonas aeruginosa, Sphingobacterium, and these are just the things that we could pick up. There's I think there's a serratia, that pink one's a serratia as well. I put this in there. I haven't talked a little bit about this, but I've done a lot of work on CPEs, and we've just recently published this year a specific CPE that we see at our hospital, especially at the Queen Elizabeth Hospital, a specific plasmid, a KPC that we only find in our hospitals, and we've labelled it, we named it PQEB. But I also found PQEB from that splash zone, so that's a CPE, an Enterobacter that had was an Enterobacter or Citrobacter that had this KPC in there as well. So you can see how you can pick up organisms. So I've talked about there is a sink splash zone. We're now even proving it that we pick up bugs from that sink splash zone. If you put filters on the tap, if you reduce down the tap flow, you get less splashing. You can do different things. If you put like a different type of sink design on there, you can get less splashing. But if you remove the filter, or you do hand hygiene or an activity, you can get more and more splashing on there. So what can you do with these sink traps? And again, that's a piece of work we're doing a bit more work on there in our lab at the moment. You probably can't see that too well. We've got actually a UV trap to try and reduce down the amount of organisms that we've got in our drain. If like our water's splashing down there, you can clearly see the UV on there, and we found that has a massive log reduction of organisms on there. You can use chemical compounds as well. So this is like a biofilm in a sink down to the drain trap, and when you put like a certain compound down there, you can get rid of it. But again, that's where it accumulates. The UV is very interesting. We found like 100-fold reduction down in like biofilm on there as well. So that was interesting. So again, that's pieces of work that will come out soon around like what organisms and some interventions that you can do to reduce that down. So for the final 5 slides, I don't want you just to think about Pseudomonas aeruginosa. I would just want you to think about all the other bugs as well. So I've talked a lot about my research on Pseudomonas aeruginosa. I've talked about CPEs and things like that. That's where my research is on. But there's other organisms as well: Legionella, Klebsiella, Stenotrophomonas, Multifilia, Cuprovidus. The non-tuberculous microbacteriums as well. There's lots of certain new hospitals have had loads of issues with non-tuberculosis microbacterium. Up in Scotland, they've had issues with Cuprovidus and so forth. And you don't need to go too far to scour the literature to find outbreaks associated with these organisms from water. Everybody knows around Legionella and the impact on that. There's people done loads of research on CPEs. Amy Matters and so forth, and Klebsiella and pneumonia. So they found like transmission of wastewater of Klebsiella to our patients. Non-tuberculosis microbacteria, as I said, like challenges and mitigations and the outbreaks of that. Cuprovidus. This is by Teresa Inkster up in Glasgow, talking about Cuprovidus. Investigation and outbreak of that, and obviously Stenotrophomonas. Loads of articles out there. I've just talked about a few little different things. So it's just some final thoughts of what else can we do? Can we use less water? So this was a lovely piece of work that was done by Joost Hopman that termed the term water-free patient care. I think it's using water. It's mainly trying to use water more safely. So this was an interesting intervention. So they looked at gram-negative colonizations infections, and they tried to go to using less water on their intensive care unit. And this is just a table of different things that they did, using more sterile water, how they wash patients. I'm not going to go through huge amounts of detail on that because I've only got a couple of minutes left. But what you can see from their study, so hopefully you can see this is colonizations, and this is just over like different time periods of like patients have been in hospital for. But the gap in the middle is when they'd use this, trying to use water more safely. And in each single one, you can see the gram-negative colonization. So this is all gram-negatives. They reduce down in every single scenario, and it's all statistically relevant. So you can see by using less water, we can see less gram-negative infections. So I've talked about Pseudomonas and other organisms as well, but gram-negatives in general have reduced down. So there's a big picture of that as well. This was some interesting work. So Giovanni Fucchini, lovely gentleman. I got him to talk at a conference a couple of years back. He's done a couple of lovely papers that he's published looking at the role of water in German intensive care units. So he's done systematic reviews and looking at it, and what he found basically in bed spaces or intensive care units that didn't have sinks in there, they had less infections when he did like a retrospective analysis. So again, showing the role of what water has in gram-negative infections and organism infections as well. Lovely piece of work, actually. In the NHS, we do a lot around sustainability as well, which is really interesting. Not a huge amount, and we have to like deliver net zero. It's a big thing, is that. But I don't know if people realise that actually wastewater and water is actually part of like the NHS carbon footprint. So using less water will have an effect of reducing that down. Or, obviously we'll use less antibiotics if we don't have so many infections and so forth. It actually it's probably a bit more than 5% of the NHS carbon footprint as well. And you can see that increase in water usage is red on our carbon footprint as well. So actually, by using less water as well, we'll be hitting our carbon footprints as well. So final thoughts, really. Hopefully you've enjoyed the talk. It's a little bit storied history of my experience, but I've talked a little bit about contamination attached with Pseudomonas aeruginosa and the transmission of that. A lot of human factors involved in that. I talked a little bit about the sink splash zone. Talked a few about other little issues of that, as well as other organisms, and I've talked a little bit about using less water. So I've done a whistle stop tour of various different things. So I hope you enjoyed it. Obviously, the acknowledgements, I am just a face, not a pretty face, of a very good team that I have who do all of that work. So I'm very thankful for all of them. So thank you.
Well, thank you very much, Mark. Yes, I mean, you covered quite a lot, but I really enjoyed certainly your talk. And usually there are no questions from the audience, but I take my prerogative as a chair to ask a few questions always for a couple of minutes before we.
No, that's fine.
And so, I mean, the sink removal has been certainly discussed for quite a number of years, but is anything happening? Because, I mean, from the evidence you provided and from those reviews, I mean, it's quite clear that it seems to work, right, to decrease gram-negative infection. So is there really in the UK or even in the hospitals a call or a drive to try to remove those sink in ICUs? But when I listen to you as well about other critical areas like your augmented care area, I'm thinking of Pseudomonas with the burns unit, is there also a call for reducing the number of unwashing sink?
Yeah, it's very, very good. The NHS, as you know, so it's like it does work very slowly, but people do recognise it. People do very good pockets of work. There's some areas that are starting to do it. So Gloucester are doing a bit of work. You've done a lot of work of like in the near units using less water. Fortunately, we've got Mike Weinbrun, who he was in one of my publications as well. I'm doing a bit of work with. He's on the new hospitals design team, mainly focusing on water as well. He's a very big advocate of wastewater, so it's certainly garnering more speed. But anything, it's like a slow moving train. It takes a while. We're looking at like moving more to like using less water on our units, but so it's getting there. It's just a slow process. So the answer is yes, but very, very, very slowly. We've got 231 water outlets on our augmented care unit. What's just far too many. We wouldn't use them all. And when you look at like the position of them behind the bed spaces, it's just silly. But anything like is a new process. So hopefully, I'm giving a couple of talks in the next couple of weeks to the NHS new hospitals program to do exactly the same as what I've just done here, shorter period of time, not an hour, a little bit around like the issues that we've seen. I'm going to go a little bit more into some of the scariest stuff around the CPs that we see. So I mentioned it briefly, but some of these like pan-resistant organisms that you find down the drain and the sinks and the wastewater, what you'd expect to see it, but now we've got direct evidence of it splashing out and what have you. That will just garner more attention.
It's always horrifying to hear you talking and other colleagues as well about the same issues.
Yes.
Removing the sink, I mean, that means you need to have other measures in place. And you mentioned carbon footprint. I know the UK is quite, I guess, leading into that. But because you have to have other measures as well, that might be more expensive, like using more wipes, for example, on other things. So is it a cost issue, or do you think it's going to be costing at the end more to remove the sink, or or actually it's going to be actually much better because you're going to reduce obviously acquired infections and you're going to save quite a lot of money, but also obviously patient or improve patient health and saving lives.
Yeah, I think it's like a happy balance, if I'm being honest, because like you can't go totally waterless because you need it, but I just think you can be better at how you do your water safe care. So I don't, I wouldn't remove all the outlets on an intensive care unit. I'll just be a bit more cautious of where I put them, and thinking about the various different factors. So that's why that design is really important. Now it is for a PFI, like a privately funded initiative. What our hospital is, we rent the hospital, so it's really expensive to remove like the taps. So you are right, it does cost a lot to do that. So it's a tricky element to do. Also, like you are right around, like if you use more wipes and more things like that, then you have like a bigger carbon footprint. But even that part of the field is progressing to have more more like degradable wipes and things like that. So I think the whole thing's evolving. Eventually we'll have like a happy medium where we've got sinks appropriately with safe care. We're not using as much as what we're doing, and we have like appropriate wipes or what have you, or appropriate factors that can fact on that. So it's like all of it's got to go together. There's a big sustainability agenda, as you said, in the NHS. What's probably similar to what's in Canada and America as well. So there's big drives on that, and they're both moving forward.
My very quick last questions is, a few weeks back we had the pleasure also to host a teleclass, which was very good, about mold remediations following extensive flooding. On part of your presentation, you mentioned you were faced with your flooding and so on. So your entire talks was about obviously all the ground negative you find as a result of wastewater and so on. Have you had any issues also with mold, especially after flooding?
Yeah. We have when we moved back into the old hospitals, when the toilets weren't used, you could see the old cisterns on the toilets, and you can see mold all over that. So you do get issues with mold. How relatable to infections that we see? We're fortunate that we don't see huge amounts of like fungal infections, but certainly that can play a role in new builds and what have you. But yeah, we do see it. We've got to I often get queries about, we see a bit of mold and what have you. But yeah, we have had that, but that's just in areas that were not used.
Mm-hmm. Well, thank you again very much, Mark, for your time on your presentations tonight. I certainly enjoy that. I enjoy every time you're talking anyway.
Thank you.
You're talking, but thank you so much for your time. I know you're a very, very busy man.
So, thank you for inviting me.
And I'm sure the audience really like that talk. And quite interestingly as well is next week teleclass is about the impact of sink removal and other water-free intervention. So obviously we quite have a theme at the moment with those fantastic teleclass all about water, on the impact of water on infection control. Here's a slide the audience can see. You've got quite a number of forthcoming teleclass, which are very interesting. Thank you, Paul, for moving the slide very quickly. Last of all is thank you very much to our patron sponsors, Diversity, Virox, on Gamma, for the support of the teleclass educations. So to the audience, it's been, again, for me, a pleasure to listen to Mark tonight. Hopefully you enjoy that presentation as much as I did, and hopefully we'll see you very, very soon to the next teleclass education. Hopefully next week when we talk about sink removal.